Hello again. Welcome back to my channel. Today we are discussing the histology of peripheral nervous system. We'll begin with the peripheral nerve. I think all of you might have seen the peripheral nerve while doing dissection, like a radial nerve, median nerve and all. It has a covering. The outermost covering is called epineurium, which is almost similar to the muscles. In the muscle we call mycium. Here in the nerve we call neurium. So epineurium is a connective tissue, dense irregular connective tissue sheath which surrounds any peripheral nerve. Inside the epineurium, the nerve fibers are arranged in fascicles. There's a nerve fascicles which is covered by perineurium. Perineurium, the thickness varies depending on the thickness of the nerve, and it contains small flattened cells which forms the blood nerve barrier. Inside the nerve fascicles we have an axon which is surrounded by a myelin sheath. As we know, all the peripheral nerve contains only the axon, and the general anatomy of the nervous tissue we know that the axon contains cytoplasm, so it doesn't contain any nucleus, so it is always do with a pink color. So surrounded by a myelin sheath. While doing a preparation of the slide, the myelin sheath will be lost, the lipid layer, so it looks empty. So the outermost covering what we are drawing outside the axon is the cell boundary of the squamous cell, and each nerve will be surrounded by a very thin layer of connective tissue called endoneurium. So endoneurium, then we have a perineurium, which is a very tough sheath. The thickness depends on the thickness of the peripheral nerve, and the outermost covering is epineurium. The nuclei belongs to the squamous cell. So if the section crosses the center of the squamous cell, we'll see the nucleus. So if the section passes through the node of Ranvier and all, we won't be able to see the nucleus. So please keep nucleus for few of the cells, not all. Similarly, the other nerve fascicle also you have to cover. So the center is the axon, which contains the cytoplasm, that's why the pink color. And axon is surrounded by the squamous cell, the boundary of the squamous cell, and you have to keep a flat nucleus for some of the squamous cell, which is very towards the surface. So bigger blood vessels are seen only in the perineurium. So very small blood vessels can be seen in the near the endoneurium, so or never seen also, because the endoneurium and the axon, they get nourishment by diffusion from the blood vessels present in the perineurium. This TS of peripheral nerve you have to differentiate from the TS of skeletal muscle. So don't make any mistake. Just observe the multinucleus in the skeletal muscle, multiple nucleus which is situated peripherally, and here the axon and the myelin sheath and the thick perineurium. Now coming to the longitudinal section of the peripheral nerve. This is the perineurium, the thick sheath. Longitudinal section, as we know, it contains only the axons, but usually when we see in the tissue specimen, the axons will be in a wavy appearance. So it is a wavy appearance of the axon. So, and we know axons are surrounded by the squamous cells with the node of Ranvier and all. So please write the squamous cell with the node of Ranvier, and so this is the H&E section of the peripheral nerve. In some of the colleges, they may show you the osmium, the special stain of the peripheral nerve, that's the osmium tetroxide. which looks black and white. So you have to draw in a lead pencil if they have shown you the special staining of the peripheral nerve. So write a wavy shape of the axon. Axon is surrounded by the squamous cell, and you have to keep a flat nucleus towards the periphery, very flat nucleus that represent the nuclei of the squamous cells and also few nuclei of fibroblast. That is the connective tissue sheath, the endoneurium, the fibroblast will be there. Make sure you write the perineurium and endoneurium, the light connective tissue inside the perineurium. That's a LS of peripheral nerve. Now coming to the ganglion. So as we all know, ganglion is a collection of cell bodies of a neuron outside the central nervous system. It belongs to two types, the sensory or autonomic. Sensory ganglion is present in all, like a typical spinal nerve. If you have read, you have a dorsal root ganglion, no? That is a sensory ganglion. It always conveys sensation towards the brain. So it is covered by a fine capsule, the thick capsule, and inside that we have a pseudo-unipolar neurons. They are the large neurons. They will be arranged in groups. The large neurons, they are arranged in groups and are separated by bundles of axons, the intervening axons and dendrites which run in between. So these intervening nerve bundles, the nerves of axons and the dendrites, they separate and forms the bundles of pseudo-unipolar neuron. So pseudo-unipolar neuron, they are large cells and they have a centrally placed large vesicular nucleus. Remember, they are centrally placed large nucleus. Surrounding each cell, we have a satellite cells. It is arranged in the form of a chain. As pseudo-unipolar has only one exit point, so the chains will be properly seen. Complete chain you can make out. These satellite cells are very squamous or low cuboidal epithelial cells. They form a chain surrounding each cell body of a neuron. And in between the bundles also you have to keep the nuclei which represent the squamous cell nuclei and the fibroblast and small blood vessels present in between the neurons, neuronal cell bodies. So this is a sensory ganglion. Remember the example of dorsal root ganglion. Now coming to the autonomic ganglion, it can be sympathetic or parasympathetic. So sympathetic is present in the sympathetic chain and the parasympathetic are present in the parasympathetic ganglion of the cranial nerves. Okay, the otic ganglion, ciliary ganglion, submandibular ganglion and all. The capsule is not that prominent as a sensory ganglion. Here we have a multipolar neurons. These multipolar neurons size is comparably smaller in size compared to sensory one. As it is multipolar, they are smaller and scattered. Okay, among the many axons in between the dendrites and axons, these cells are smaller in size and scattered. They are not in groups, they are scattered. And they have a round large nucleus, but it is peripherally placed. Remember, in the sensory we have centrally placed, here we have peripherally placed nucleus. And as it is a multipolar neuron, you have a lot of processes, the dendrites, axons and all. So the chain of satellite cells will be broken in between. So you won't see that much prominent satellite cells here. Satellite cells will be present but not that much prominent as sensory ganglion, and you have to keep the nuclei for fibroblast and the squamous cells. So make sure you know the difference between the sensory and autonomic ganglion. I hope the video was helpful. Please like, share and subscribe to my channel. Thank you.